Hello, good morning. Here Dr. Nagin Kumar Chandravanshi. Today I am going to discuss about antisense technology. So this is the content from molecular biology MSc second semester biotechnology. So this lesson will cover general introduction, molecular mechanism of antisense molecules, type, generation, mechanism, and its basic function like inhibition of splicing, inhibition of polyadenylation, inhibition of translation, and their applications. So you can see this is the overview of antisense oligonucleotide development. This is just overview. It has started from 1978, and then this is going on step by step, and there are number of different challenges which have already optimized in different time by different researcher, and now this most of the developed antisense molecule drugs already in clinical trials and many of in the market. So now we are going to discuss about introduction. This is the basic idea about antisense oligonucleotide molecules. So what is antisense? Antisense basically is oligonucleotide compound materials. This is the genetic material is a single strand RNA which act as antisense. So first time it was discovered by Jackman and Stephen in 1978, and then later this technology again optimized by different application, and nowadays it is used as a powerful tool for target validation and therapeutic purpose for number of different genetic altered molecular disease, and nowadays it is used for inhibition of viral infection, cancer growth, and inflammatory disorders. So this technology is having a huge number of challenging, so still work going on. So now we are going to talk about types of antisense oligonucleotide technology. So this is basically three different type. You can see this is a type of DNA, and they are ultimately what in the central dogma procedure. DNA transfer in mRNA, that is called translation. This is about transcription procedure, and then mRNA again transcribed in the final development, that is about protein development. This is called translation. So in case of the healthy DNA, healthy body, what they could produce healthy mRNA, and now they're developing us protein molecules, strong protein molecules. But sometime they are genetically affected, altered by mutation. What about different region? Now this one developing some altered mRNA, and they are definitely will produce some altered protein. They may be have hampered to human health, and so this is not important for our human health, the regulatory mechanism. So it should be stopped by some activity. This is called antisense, because you can see DNA is developing from two different strands. One is a sense, another is antisense. So DNA first mRNA transcribed from the antisense strand. So this is called so now we have to develop antisense molecules. So you can see this is about antisense oligonucleotide, and the second option is ribozyme. Yeah, we can say DNA enzyme. So ribozyme is what? A small number of different ribonucleotide element in our cytoplasm, huge number of there, and the different category. They are directly bind to target mRNA, and what it can cleave the small of mRNA, and that's why the particular mRNA cannot translation in the next phase, and there is no protein development. So this is optimum stage of antisense technology. And in the next category, small antisense RNA. So number of small amount of mRNA, ribosomal RNA, siRNA, snRNA, they bind to directly target mRNA, and ultimately what they developing RISC, this ribosomal induced silencing complex, and what they stop the activity of translation purpose, they inhibit the activity. And you can see here, and simultaneously what the particular antisense oligonucleotide molecules, they also stop the capping, polyadenylation, pre-mRNA processing, splicing, post-splicing, stop the translation activity. They stop the ribosomal binding site, the inhibition of inhibition of translations, and there. So this is a different category activity done by antisense molecules. So in the next slide, so we can see, so just we can compare with antisense technology and the conventional drug. So what about in antisense agent? This is very more powerful because they directly bind with the cellular level and they stop the translation in ultimately development of the protein. So this is more effective. And ribozyme and DNA they are categorically active oligonucleotide. They only bind, but they cleave the target mRNA. But some strong evidence also prove that most of the antisense technology will again rectification by developing to higher nucleoside degradation and enhance target affinity. So this is the major development in antisense oligonucleotide technology. But siRNA is also having good activity in terms of the suppression of undesired gene suppression. So nowadays this technology used in different animal and other experimental activity to find out best suitable way to suppress the activity of undesired element. So at early stage widely used knockout technology. So knockout technology they are having a lot of problem, like sometime it is very expensive, it having a labor intensive, sometime there is no cases, sometime already experimented had done, but due to the lethality, yeah, some activity finally failed the final result development. So this is the major challenge in classical technology, yeah, conventional technology. But antisense technology is very attractive technology that actually suppress the target gene. And what the again advantage antisense having some molecules we can design after the development of human genome project. Already we have a sequence are there, so it is easy to develop synthetic oligonucleotide and molecules that is directly delivered to body, and it may good interaction and good with having higher nucleolytic degradation resistant activity plus higher affinity. So that is good advantage for antisense molecules. In the next slide, you can see what is the basically antisense oligonucleotide. It's about 15 to 20 nucleotide which are complementary binding, and there are two different activities there. You can see the first RNase cleavage activity you can see here, and the second one is a blocking of translation. So what happened? Antisense molecules directly bind to the target mRNA. You can see here, and what its interaction they promote recruitment of RNase H, ribonuclease H. H is hydrolytic enzymes, and then cut the target mRNA in presence of binding oligonucleotide. So this is the one important activity of antisense molecule. Another thing is what? But sometime there are different antisense molecules there. So this is the generally done by RNase H activity, but somehow due to the modification generation wise development of oligonucleotide, sometime not properly working, and so that time blocking of translation, that is important concept for antisense activity. And you can see when antisense oligonucleotide bind to the mRNA and there is no ribosome binding site, and when failure of the ribosome binding the particular mRNA, there is no translation is there. So this is the what? This is the just suppression of protein expression, that is called central dogma is failure. So this is another important approach for antisense oligonucleotide. So you can see this is the already we have seen, but it's very important slide to just summarize how to work. And so this is basically done by different mechanism. You can see here HDS RNA, double strand RNA. So double strand RNA is nothing else but it just bind with complementary RNA to target mRNA, and what ultimately developing siRNA. So siRNA means single this is what interference RNA, and now developing in RIC, ribosomal induced silencing complex, that promote always to clip, it's like clip the particular phosphodiester bond between two different nucleotide, and what our the particular nucleotide will cut in different element. So this is what this is the this cleavage activity. This cleavage activity stop the gene expression, yeah, translation purpose. So they ultimately stop the activity. And now we are going to in this slide we are going to discuss about type. So how to use the different antisense molecules. So they are used in different terms. The first, how to use, we have data of different nucleotide. They are already developing, they already develop in having the different biological databases, nucleotide databases, that is called bioinformatics in silico tool technology. So presence of this technology we can apply, and we can develop some design oligonucleotide. So oligonucleotide basically it's a type of some nucleotide bases about 5 to up to 10, sometimes 20, sometimes 25. So this is the type of developing, and you can see this is used in different parameters. You can see the first parameter is called non-random. Non-random means what? Strategic base. That means this is the target mRNA, and already we have know the idea about this particular the segment, and now we have to develop the exact copy of for particular target mRNA. So this is called non-random. Now that time they directly bind and stop the protein activity, protein translation activity. So second thing is random. So random is basically is what random means. Sometimes you just assume here a u g c some segment like a g u c, but sometimes like a we are developing the complementary like t here u, here a, here like g. So this is just random, and that time promote based on RNA's activity. And we have seen in the previous slide RNA's activity directly, this is the target mRNA, this one, so directly cleave here. Okay, so there is what, this is the based on RNA's recruitment activity. And again semi-random means some component like up to 50 to 60% they are directly higher affinity with target mRNA. Now this time maximum binding, maximum complementary binding, Watson-Crick binding, and that directly promote to RNase H. So what, they directly cleave in the small segment. So this is the another experimental activity of uses of antisense oligonucleotide for the experimental purpose. So in the next slide, we are going to discuss about how to design. So you can see in our nucleotide there are different nucleotide A, U, G, and C. Okay, we are going to discuss about mRNA. So there is no T in place. In place of the T there is a U. So you can see we have to focus always G residue. So what about when antisense developing, in case G is there, so they are making triple bonds there. This is called Augustine base pair. So it should be avoided. And the second thing is what? CpG. And as we know that in our genome, this huge amount of CG isolate is there, huge number of. So they directly promote to immune stimulation activity. So sometimes this is the limitation of some antisense molecule. So we have to keep in process of development design already avoid to G residue development and CpG motif. So that's why our antisense molecules is much better and directly higher affinity between target mRNA, and stop the expression, including they stop the nuclease resistant activity, enhance the life cycle. That is good for designing purpose. The second objective is what? Now we are going to discuss the modification. So modification is very important. What is the important modification? So modification is that is simply simple normal nucleotide just transfer in host body, either purpose for treatment. So sometimes normal nucleotide they directly recognized by different nuclease DNA enzymes, and they will cut, and so what, the drug is not directly antisense molecule not directly bind to target mRNA. So our purpose is completely there is no solve. So that means what, we have to change based on the analog basis, like number of different bases there, like adenine, guanine, cytosine. So we have to change some similar analogous bases, and we can add it some unnatural. Similarly, we can modify sugar. So sugar backbone is very important. We can modify it here, and similarly we can alter the phosphate backbone. Phosphate backbone is there is some free oxygen is there. So directly we can replace, and we can modify it. What about our different suitable element like some elements, some metals contain. So it may be much better to enhance the life cycles of particular antisense molecules. And so you can see here binding of antisense oligonucleotide. So another thing is what we have always avoid sequence motif associated with non-antisense molecules. We have to directly select some individual known higher affinity sequence. The second thing is what treatment of rodent some primate in this is the. So I'll develop CPG. This is the immunostimulation. This is the challenge for management of antisense drug delivery. The next point, string of guanosine, that already we have discussed. G residue that promote no antisense activity. जहाँ जहाँ पे G binding is there, they make histone base pair, and that negative feedback for particular antisense molecule. And the next point, identification a potential mismatch site. Sometime mutation happen in our genome, but and we are already going some developing a drug. So our target mRNA should be known. So that's why develop synthesize antisense molecules is higher affinity, and higher affinity is promote silencing stop the activity. So this is done by FASTA and BLAST, another process. Avoid site of polymorphism. So polymorphism is a big challenge for antisense oligonucleotide therapy. So we have also keep in mind always avoid site of polymorphism. Avoid cross reactivity sequence. Cross reactivity is a big challenge. Similarly, some potential aptameric motif is there. We have also avoid. So this is the major challenging and limitation restriction point keep in our during the process of development. So already we have discussed the modification in the different bases. You can see this is the structure of ribosome nucleotide, and you can see here base. You can see here 2O regions, here phosphate region. So what in basically we can change our bases. Some 8 base analog we can change here. Sugar backbone we can change here the phosphate region. So it's about what normal nucleotide. It can enhance the life activity. It can prevent it from interaction with already nuclease and other ribonucleotide enzymes. So you can see this is the category of antisense molecules. We can discuss in upcoming slide in step by step. The first will cover first generation, then we'll go for the next second generation, then third generation. So this is about the first generation. You can see the first generation. What about we have discussed? The first we can change our bases. We can change our 2 region. We can change here this phosphate backbone region. So now you can see this is this is the first generation antisense molecules, and now you can see this is the as sulfur. So this is the first generation antisense oligonucleotide. The free radical oxygen will replace by as sulfur. So now this is called phosphorothioate DNA. Why DNA? Because at early stage of the target mRNA, which directly bind by this particular antisense molecules. So this is called and look like just complementary pairings, something like that. So this is called antisense oligonucleotide, and this is the first generation antisense product. So this is about some antisense first generation molecules. It is first synthesized by changing the non-bridging oxygen. Already have discussed. And what is the impact? It can more resistant. You can see this line. This one more resistant to nuclease. And what if they are resistant? They directly increase the half-life of particular compound. So this is very important. And the second thing is what it can synthesize the negative charges. As is negative charges, and phosphate we know DNA is the region of due to the region of phosphate they are negative charges. So already sulfur having the negative charges. So the easy to delivery cell delivery is more important thing that directly promote pharmacokinetics. In case less number delivery, not targeted delivery, that there is challenge for the antisense molecule. But in presence of this phosphorothioate, they are having negative charges, so it easily delivered to target region. and this is the first this is the only FDA approved drug for trial and other thing. And but they are having some issues like some side effect in vivo trial cases, like immune stimulation and complement activation. So what this is the some drawback for host body. And another antisense in the first generation, you can see phosphatide we have seen, this is called PS, phosphatide. Another antisense molecules, that is this is a methylphosphonate, this one, and you can see this adding CS3 group, and this is again, this one is phosphorimidate. You can see NH is just like bridge between sugar and phosphate backbone. So this is the first junction, but this is the phosphatide is the most highly preferred antisense molecules in the first generation. And already we have discussed this about this is the FDA approved, this is called Vitravin. So Vitravin is a first generation antisense molecules. This is basically useful for therapeutic uses like cytomegalovirus infection, and is caused by HIV and promoted, and in a pharmacotherapeutic groups like ophthalmological application. So but and this is the some branded name Formovirsin and common name Formovirsin. So this is the first FDA approval drug for treating to some type of HIV infection develop cytomegalovirus meningitis infections. So now we are going to discuss about second generation. What is the change in the second generation? So what is the important objective of the second generation? Especially what is the first generation change here, thiode group, but this was replaced by AS chain. But uh, so the next generation change here adding CS2 in the OH region. So this is known as a 2O methyl antioxidant antisense oligonucleotide. The second thing, you got 2O methoxyethyl. You can see OCS2. So this is another antisense molecules in second generation. But there is some drawback in terms of development of RNase-H recruitment. Already we have seen RNase-H recruitment is very important in terms of for recruitment for cleavage activity. But this one this one makes some altered structure, and this altered structure stop the further activity like transcription activity. So this is some challenge for the second generation. So this challenge again resolved by GAPMA technology. So GAPMA technology is technology that you can see, just this is an example. This is the target mRNA, and what in case of simply modified, this make some altered structure because there is a presence of the structure of two origin. But this one modified by adding of GAPMA technology. So GAPMA technology, what it can enhance and interaction with particular higher affinity of mRNA, and it is also promote maximum uptake by our tissues. So this is the good. And another point is what longer in vivo half life and laser toxicity. This is the important of second generation with GAPMA technology. So GAPMER is what? GAPMER is, you can see this is the nucleotide is there, and some nucleotide from hybrid from the first generation and some from second generation. So you can see this is about the second generation, and the first line about this is first generation, and this one second generation. This is the first generation. So what it can create maximum interaction between target mRNA. So you can see this GAPMA technology. This is the target mRNA is there, and what the GAPMA technology is what directly bind with the target mRNA and promote RNase activity. But only second generation molecules it cannot able to promote a recruitment of RNase activity. So this is the challenge for second generation. But presence of the GAPMA technology it is resolved, and this is much better than primary generation, a first generation nucleotide. So some example you can see here. This is the five. This about this about the five, and this is, three prime wings. And what about changes? the most important changes you can see here. The five, this is 14 mer, 14 oligomer, and this is especially for target. This is developed for, and some element from first generation, and some element, some nucleotide will modified like a second generation. So this is the target for PTN. This is causing some type of mutation that important for the cancer. So this is the another application of GAPA technology. And already we have seen interaction with antisense oligonucleotide. They stop the capping, the start of polyadylation, also including the stop of splicing factor. We have know already most of the exon and intron is there in our eukaryotic genome. So after the post RNA processing mechanism, splicing is one mechanism, and spliceosome bind there particular region. So presence of the antisense molecules, they stop the splicing activity, and what then this one promote like some undesired type of activity, and this is a not proper modification for translation activity. So this is always drawback for protein suppression, yeah, gene silencing activity. So now we are going to discuss about the third generation antisense molecules. So already we have seen the first generation discuss about here, the first phosphate chain, second generation is here, and now the third generation. The third generation is what is much better than first and second generation, because they directly bind to do DNA back to like RNA, but acting as like DNA, double strand binding like. So here modified sugar backbone chains, and similarly interaction similarly change the logged nucleic acids. Some sugar molecules already directly to logged, sometime added up some nucleotide, that is called peptide nucleic acids, sometime used, you can see modified here NH group, sometime 2-fluoroamino acid are added here, sometime you can see this is a logged nucleic acid. So the different oligonucleotide developed in the third generation. So here is what ultimately change in the furanose ring of, yeah, we can change sugar ring, we can change in the phosphate linkage. So that is very important for the third generation, and the entire modification directly promote to higher nuclease stability, higher target affinity, and now they ultimately what a pharmacokinetics profile of the oligonucleotide. So here mostly well known terms like logged nucleic acid, peptide nucleic acid, and the morphinophosphoroamidate nucleic acid. This most commonly in the third generation, and huge number of oligonucleotide there in the third generation, but only 3 or 4 nucleotide is much important due to their higher affinity, higher application in vitro and in vivo experimental activity. And nowadays this is the reason behind that most of the clinical trials go in a phase second, phase third, and many of are already approved by FDA. So this is what you can see. This is another application. Peptide nucleic acid. This ability to recognize DNA, and they are capable to modulate some gene expression and inducing mutation. So this is very important for the third generation oligonucleotide. And you can see that here different categories of oligonucleotide third generation, and this is about the peptide nucleic acid. You can see basically nucleotide is look like, and they make directly interaction, and they make a bond, but in presence of this type of backbone of sugar, ultimately what this is altered, sometime altered, and this problem this interaction is completely non-Watson-Crick binding, and this not Watson-Crick binding at what more stable, and this is more nuclease resistant activity. You can see this is about the log nucleic acid. This is the log. What about the furanose ring is completely logged. So now this structure is more stable in a higher nuclease medium. So this is about some concept for antisense third generation molecules. Already we have discussed in previous slide. So now this is the overview of antisense mechanism of action. Now you can see this about what in case of the fourth generation, in case of the second generation, in case of the third generation, they ultimately what they bind to the target mRNA and recruitment of the RNase H. So RNase H is what presence of the RNAs cleave in the two small different fragments in the particular target mRNA, and this particular oligonucleotide is directly target a specific region, and they promote binding and with higher affinity binding, and uh, this binding recognized by RNase edge. So RNase edge what just cleave in the phosphate in between two different nucleotide. So this is the one important term. The second thing is what, when interaction oligonucleotide in particular initiation region, you can see the cap is there is no cap development. They also promote to translation arrest. So there sometime this is the coding region, you can see, and this is the end region. When splicing, when antisense bind there, and when antisense bind in terms of half segment of exon and half segment of intron, they directly affect to interaction spliceosome machinery system, and this promote to antisense silencing activity of targeted mRNA. So the next point is very important point. Now antisense molecules are developed basically in some particular terms, and now how to deliver. So delivery used for different matter like mostly important transfaction region, and the second way it may be lipofaction technology. Some research laboratory and research group already working in a region, and the next is what, some unassisted uptake is also possible due to the some gapmer. This is the gapmer is bigger size. Normally oligonucleotide is about 20 up to 15 to 20 oligonucleotide, but gapmer is just having a huge size, so sometimes use is very challenging to uptake. So this technology, this developed methodology will manage by different synthesizing process to deliver like positive gapmer control already delivered in the host body for the clinical trial and check their validation, and similarly some negative control also use that time, and then recognize their activity. What about the affinity, what about the nuclease activity, what about the half life, and what about the protein expression activity. So this will check by using some control parameter. And gapmer technology also check by some fluorescence level element, because the gapmer is just designed between it may be first generation and second generation, it may be third generation and the second generation. So the gapmer is just hybridone technology for two different nucleotide. So now you can see this is different type of antisense molecules are developed, and they are transferred in the different ways. Some it's about the targeted region, use some different region. You can see this is the intrathecal CNS motor cortex. So here use different technology intrathecal administration in case of the rectal administration, in case of the intradermal skin transfer skin, in case of the aerosol for the lungs, in case of the intravitreal eyes. This is the different delivery approaches based on the targeted problem. So how to deliver particular antisense, this is a challenge, and the particular region, this is a big challenge. And still going this is the current this is the very height this is the very highly demand scientific research. This promote personal medicine. So nowadays different type of antisense molecules already trial in the different generation, and you can see I have shared some research paper review articles. You can see that this for systematic and some local, and this is the different delivery approaches there. And here you can see this is a phosphorothioate antisense molecules, the different name this developed from different companies there. And this is the some disease, and this is the chemistry how to interaction, and what about the interaction is there. And you can see like mipomersen, this is about PS2MOE. So P as phosphorothioate and MOE, this is the second generation of and first generation. So this like just gapmer, this one again gapmer produced product. You can see this is first generation and the second generation product oligonucleotide. And this is what I'll this is about phosphorothioate oligodeoxynucleotide. This is the very important for rectal administration region. This is for the local delivery approach. And now you can see this is the delivery of particular antisense molecules. What about in our host body, your recipient body? You can see this is antisense going inside in the different region, and there's just start interaction with target mRNA, and the time bound activity, they cleave small molecules, and if they are higher resistant against the nuclease activity, they will move in the next generation. And you can see this is the cytoplasmic activity and this is the nucleoplasmic activity. So the different time activity that enhance their capacity of interaction with target mRNA, and ultimately that destroy the particular target mRNA, suppress the activity. So this is the delivery mechanism is very important for develop and synthesize oligonucleotide. And this is about some number of different antisense molecules now in the market. Is in market, you can see the veterans. This is about developed from ISIS pharmaceutical company and targeted for CMV. Already we have seen cytomegaloviruses, and this promotes meningitis disease, and the chemistry is what? PS means first generation phosphothyroid DNA means directly bind with complementary bind, and now this is FDA approved. And this is the list of the category of different antisense oligonucleotide you can see here, and the different branded companies, they are having some brand, and this is target specific activity, and this is about particular disease like cancer. So GTI 2040, this is developed from Loris Therapeutics, and now this is especially for the cancer disease, and the region and type of antisense molecule, that is PS DNA, and now this is a phase second trial. So most of the antisense molecules is going in trial, and maybe in the near future they're coming as available for the particular personalized uses for treating to varieties of disease. Similarly, you can see this is a number of different examples there, and this again available in different trial phase. So this are the references. So already we have discussed what about the antisense molecules, how to design, and what is the generation, and what is the limitation is there. So what is the challenge? So and how to use most optimized, highly efficient oligonucleotide. So it will develop by already developed bioinformatics laboratory with nucleotide bases, and so but sometime random and semi-random that challenge to manage, because it optimization and delivery is very important. So for the targeted treatment, you need completely non-random. So that's why, and we have to know exact procedure of delivery in particular host body and host tissues, affected tissues region. So then it can move in the particular region and will bind to high affinity and more resistant against nuclease enzyme. Then that's basically objective of antisense molecules. And number of different research laboratory in worldwide, they're going research on. So in future maybe they have good product for personalized medicine, that is also called molecular medicine. Thank you for